Thank you for the opportunity to discuss some of our ongoing research on the gut microbiome here at ISB, particularly how it pertains to statin and personalizing statin therapy. So statins are a cholesterol-lowering medication that's proven highly effective in decreasing cardiovascular mortality risk. About a quarter of adult Americans are currently prescribed statins, with some of the most common brands out there being Philipid, Zocor, as well as Lipitor. Although statins have proven highly effective in decreasing cardiovascular risk, their use does come with several side effects. Some are minor, such as headaches or nausea. Others, such as myopathy or muscle pain, actually leads to discontinued use of statins among some users. An additional common side effect, although it's commonly reported, it only affects a minor proportion of individuals, is an increased risk of type 2 diabetes. This is one plot demonstrating the concept throughout the course of 8 years of follow-up. The blue line is the incidence of diabetes in non-users, while the red line demonstrates the progressive incident diabetes across 8 years in statin users. While the risk of diabetes alone isn't necessarily encouraged to discontinue statin use, it is a worth side effect to try and mitigate, to improve patient adherence and also mitigate any side effects and really benefit from the drug as a whole. In addition to side effects from taking statins, there's considerable heterogeneity in how individuals actually respond to statins in terms of their on-target effects or lowering LDL cholesterol. This is a figure we'd like to show from a randomized control trial, the Jupiter trial. All of these individuals were prescribed or given the same dose of rosuvastatin. And on the y-axis is the percent change in LDL cholesterol from baseline and then upon initiation of the treatment. While about 50% of individuals achieved the desired 50% reduction in LDL cholesterol, you can see that another big proportion of individuals achieve a much lower reduction in LDL, while some individuals actually see an increase in LDL while on the drug. Why this is particularly important is because the extent to which statins can decrease LDL cholesterol directly corresponds to your risk of developing cardiovascular disease risk. And that's shown in the box, in the bar plot to the right, where individuals where LDL decreased the most also had the most benefit in terms of decreasing cardiovascular risk. So we really wanted to ask this question, is the gut microbiome involved? We see this heterogeneity both in on-target and off-target effects of statins. Does the gut microbiome play a role, and is there a clinical translational component to it that can be utilized in the clinic? And there's some experimental studies that point that this might be the case. Statins and other drugs are widely metabolized by gut bacteria. One example is from a recent Nature paper where in this particular graph on the y-axis, you see the percent of the drug consumed, in this case, lovastatin. And each of these bacterial isolates was grown in vitro, and the drug was given in culture. And 48 hours later, the amount of drug left was measured. And you can see there's a number of different gut bacteria that are able to metabolize 50 or 80% of the statin that's given to it in culture, indicating this broad and diverse metabolism of statins by the gut microbiome. Statins have also been shown to alter gut microbiome composition. And since the gut microbiome directly contributes to metabolic health, glucose control, and inflammation, there's multiple different mechanisms through which statins can impact metabolic health and can be modified by the gut microbiome in terms of their on-target effects. So the main question we wanted to answer is, can gut microbiome composition explain this wide heterogeneity in statin responses? To tackle this question, we utilized a cohort of individuals, close to 2,000 individuals with what we call deep phenotyping. These individuals had their blood drawn and had an on-target metabolomics measure on their plasma. In combination, they also had clinical lab measurements, extensive interviews on their prescription medication use, as well as gut microbiome data on a large majority of them, 1,500 individuals. Not only do we have information on whether individuals were on statins, for a subset of individuals, about 90 or so, we also had detailed information at the intensity of the statin prescribed, high, moderate, or low. So this gave us a nice platform and a nice data set to dive into some of the questions we had about statin responses and the gut microbiome. Since we didn't have pre-statin LDL cholesterol levels for any of these individuals, all these individuals came to the program already on statin, we had to identify other potential biomarkers that could help us gauge how effective statins are in inhibiting their target enzyme. On the left, you see a simplified version of the de novo cholesterol synthesis pathway that statins target. They inhibit the rate-limiting enzyme of this pathway, HMG-CoA reductase. And one of the first things we observed is that statin users show a markedly higher level of HMG in the plasma, shown here highlighted in red as well. And this made a lot of sense since HMG is the hydrolyzed product of HMG-CoA, which is the substrate for the enzyme statins inhibit. But not only did statin users have higher levels of HMG also showed a negative correlation with LDL cholesterol, but exclusively in statin users, indicating that HMG not only reflects statin use, but also it reflects the extent to which the drug inhibits an enzyme in a cross-sectional design like ours. As further sanity checks that HMG is indeed a reliable marker, we looked at the association between HMG and statin therapy intensity, and that's shown in the box plot to the left. And we saw a nice dose-dependent curve where the higher your statin intensity, the stronger statin you take, the more HMG buildup there is in the plasma. In addition, there's been several large-scale pharmacogenomic studies identifying genetic variants that predispose individuals to different responses to statin therapy. So we evaluate if HMG actually captures some of that genetic signal. And this is one example on the right where this is RS7412 SNP within the ApoE gene. As you can see, minor allele carriers show no difference in HMG among non-users on the left. But the moment individuals get on statins, you actually see the shift where people with the minor allele have a stronger response in terms of HMG. And this is very consistent with what's been shown in randomized control trials where individuals with this genotype respond better, have a lower LDL cholesterol at the end of treatment. Next, we shifted to the gut microbiome. And initially, we just wanted to confirm previous studies showing that there's an association between statin use and gut microbiome composition, focusing on this large kind of structure metric, which is alpha diversity, how diverse is your ecosystem. We did observe some significant difference, although very minor, in terms of how statin use is associated with the gut microbiome. Sham diversity on the left showed a significant but very modest decrease among statin users, which is consistent with some animal studies. We also looked at statin intensity therapy, which is the bar plot to the left, but we didn't observe any dose-dependent decrease in alpha diversity with increasing statin intensity, with only the moderate intensity group of individuals showing a significant decrease, while the high intensity was actually trending higher than the non-user group. And then we started asking the questions we were really interested in, is mainly, does your gut microbiome composition when you're on statins correspond to how well you respond to statins? And we did this through interaction analysis framework. In this case, we modeled HMG, this marker of statin effectiveness, and we included alpha diversity by statin interaction term. Mainly, we wanted to see associations between gut microbiome and statin use that are only present among statin users and markers of effectiveness. And what you see here, and these are two different alpha diversity measures, sham diversity on the left and observed ASVs, or the number of species present in your gut ecosystem. As you can see, as the gut microbiome becomes more diverse, the increase in plasma HMG in statin users relative to non-users decreases. So people with very diverse microbiomes tend to have lower HMG than people on statins who have very low diverse microbiomes, indicating there might be some involvement of the gut microbiome and how statins work on this target enzyme. We focused next specifically on individuals where we had detailed information about their dosage therapy, and we adjusted our models for sex, age, BMI, and this dosage intensity variable. But what you can see in the red plot, scatter plot, even after we adjust for all these covariates, there's still this negative association between HMG and alpha diversity, indicating that individuals with more diverse microbiomes have a less on-target effects of the statins, have a lower decrease in LDL cholesterol, are less responsive to statins. As a sanity check, we compared all of the same analysis in non-users, which are the blue points below, and there are no associations between HMG and alpha diversity measures, kind of giving us stronger evidence that whatever's going on is dependent on statin intake and only emerges among statin users. We further looked at LDL cholesterol, and oftentimes there are target goals that clinicians want to reach with their patients who are on statins. And depending on your risk of cardiovascular disease and other comorbidities, this is set to either less than 100 milligrams per deciliter or less than 70 milligrams per deciliter. And you can see, regardless of which alpha diversity metric we use, and we adjust for a number of covariates, including sex, age, BMI, type 2 diabetes status, individuals with more diverse microbiomes have a lower likelihood of having reached this target LDL cholesterol level, providing more evidence that a more diverse microbiome might interfere with statin on-target effects. Shifting a little bit in how we think of the gut microbiome, right now we've been mostly talking about these higher order measures of alpha diversity. But there's also a concept of enterotypes, where certain clusters of microbiome composition an individual can fall. These vary based on population, but generally in the Western world, in Europe and the United States, there's 3 or 4 main clusters that your microbiome composition may fall into. And there's been some previous reports showing that these clusters actually correspond to statin therapy as well. So these enterotypes shown on the upper left here are either 2 Bacteroides enterotypes, where the dominant microbe are species within the Bacteroides genus. The less prevalent in the Western world Prevotella enterotype, as well as a Firmicutes alpha diversity rich Ruminococcaceae enterotype. And previous studies have shown that obesity tends to correlate with these enterotypes, with BAC2 enterotype having the highest prevalence of obesity, while the Prevotella and Ruminococcaceae enterotypes seem to have lower prevalence and tend to be healthier in general. So we initially want to replicate these enterotypes in our own cohort, the Arivale cohort. And this is shown here, kind of a broad figure. It was a PCOA analysis, or principal coordinate analysis. Each microbiome profile is projected into 2-dimensional spaces. And further validating this through statistical modeling, we actually observed that there are indeed 4 distinct enterotypes in our cohort, and they correspond very nicely to previous studies. We have 2 Bacteroides dominant enterotypes, shown here on the bottom by their relative abundance of Bacteroides, 1 Prevotella dominant enterotype, and then also this distinction between the BAC1 and BAC2 enterotype, shown here on the far right of the slide, since BAC2 enterotype is also associated with lower prevalence of commensal short-chain fatty acid producing taxa, like Faecalibacterium. So having this enterotype, we next once again want to investigate whether your membership in a specific enterotype dictates or corresponds to how well you will respond to statins. Looking at LDL cholesterol, and this was modeled using ANCOVA with the interaction term, as well as adjusting for multiple covariates shown on the bottom. We observed that the BAC2 enterotype in particular had the greatest response or greatest change in LDL cholesterol among statin users relative to non-users within the same enterotype. If you look at the gray box plots, which are non-users within each enterotype, you see there aren't very many differences or very strong differences in LDL cholesterol. But the moment individuals are on statins, you see all these shifts, which are further shown in the bar plot to the right, where the BAC2 enterotype really sees the greatest decrease in LDL cholesterol. We next really want to focus on side effects of statins, and in particular, this concept of type 2 diabetes and disrupted glucose control. So we focused initially on HOMA-IR, which is it's an index that reflects insulin resistance. It's a combination of blood glucose and blood insulin. And once again, we run our statistical models with the interaction term, adjusting for a number of covariates. And on the y-axis here is the log-transformed HOMA-IR measure adjusted for these covariates. And what's really remarkable is that if you look at statin users, while there's a significant increase in markers of insulin resistance in the BAC1 and BAC2 enterotype, individuals with the Ruminococcaceae enterotype show virtually no difference, indicating there's no risk of metabolic complications in individuals with this microbiome composition when they go on statins. We expanded this analysis into multiple other markers of insulin resistance, blood glucose, blood insulin, as well as glycated hemoglobin A1C. And in each case, we see the same associations that remain significant after adjusting for multiple covariates. The 2 Bacteroides enterotypes show these pretty profound increase in markers of insulin resistance, while the Ruminococcaceae and Prevotella enterotypes really show no significant differences, indicating that your gut microbiome composition may dictate your risk of metabolic complications. The question that remains is, why are these Bacteroides enterotypes at higher risk of metabolic complications? And there's several possible explanations that we hope to answer in the near future. One is that Bacteroides species, and I've touched upon this a bit earlier, are the major drug metabolizers in the gut microbiome. And this is mostly from in vitro experiments, but on the y-axis here, you have the number of different metabolized drugs for each bacterial isolate. The red bacteria are all within the Bacteroides species, and you can see these are some of the greatest drug metabolizers within the whole microbiome. This may result in greater activation of the prodrug, or it can also result in modification of the statin by gut bacteria, contributing to potential side effects of the drug. So in summary, microbiome composition seems to significantly modify associations of statin use and markers of both statin effectiveness, on-target effects like LDL cholesterol, but also markers of insulin resistance. I'd like to point out that a major limitation of our study is its cross-sectional design. Although we were able to adjust our models for a number of potential confounders, we weren't able to actually get the pre-statin LDL cholesterol level, which would give us the best insight on how well statins work in the individual. The ruminocaccase enterotype seems most protective from adverse effects of statins on glucose homeostasis. And there's some logic to it, since this is the enterotype with highest alpha diversity, and also abundance of Akkermansia, which is an anti-inflammatory microbe, as well as certain short-chain fatty acid producing taxa, which are known to positively impact human metabolism. Bacteroides enterotypes, on the other hand, show the highest efficacy. Individuals with this enterotype seem to respond the strongest to statins on-target effects, but also increased risk of metabolic complications. So one potential implication is that individuals with this enterotype could benefit from a lower statin dose, which would still allow them to achieve their desired % decrease in LDL cholesterol without the side effects that come with it. Additionally, we could start thinking about complementary probiotic and prebiotic interventions in these individuals. However, further experimental studies are needed at this point to really understand the mechanisms behind these associations. With that, I'd like to quickly acknowledge all our funding sources, the NIH, the Washington Research Foundation, and my PI is Nathan and Lee Hood, as well as our close collaborators, Sean Gibbons and my close coworker, Noah Rappaport. Thank you very much.